I'm gonna talk in genomics and cancer. So the context of this talk is first I'm gonna give an introduction to the talk. Then I'm gonna talk about a little bit of cancer genomics and next generation sequencing. After that, I will talk about variant classification, like germline variants and somatic variants, the difference between driver and passenger mutation, and the variant classification by ISCB-LA. Then I will focus in breast cancer as an example of cancer genomics, and specifically germline variants in breast cancer. Some studies that I have worked in Guatemala and Latin American consortium. So then I will talk about other cancer syndromes that we have found in Guatemala and the conclusion. So I will start with this phrase: cancer is a disease of the genome, because cancer is an acquired genetic disease, and is caused by the presence of genetic alteration. Some genetic alteration could be DNA change, epigenetic alterations, chromosome alteration, and changing gene expression. But it's an acquired disease because it's caused by, the majority of time by environment factors. So, for example, we know that smoking influenced the development of lung cancer. So I participated in a course of cancer prevention by NIH, and I received a talk about nutrigenomics in cancer, and they talk about how some foods could predispose us to colorectal cancer and stomach cancer. For example, eating vegetables and fruits avoid us to develop a colorectal cancer. But if we eat too much desserts, we could have more colorectal cancer. So I was thinking that everything I eat could cause me cancer. So, okay. So cancer usually is caused by accumulation of genetic alteration. It starts with a pre-lesion in a tissue, and then it proceeds to an in situ cancer, and finally to metastatic tumors, and then could be lethal. And cancer is a dynamic condition. So this means that the genome is always changing. So that's the difficult to find new treatments to treat to cure cancer. So we could find a lot of, as I told you, a lot of alteration. We could see mutation, the DNA change. We could see copy number variation, structural variation, as chromosomal variation, and changing RNA expression, and genetic alteration. And why is the importance to study mutations in cancer? First, this could help us to study the classification, the molecular classification and histological classification. And then, for example, I could use the sequencing of genes to study resistance mechanisms, or to select the correct therapy to some cancer. And other application is to study the genomic of metastasis. In this case, we could find some genes that indicate progression, indicate the metastasis in a patient. So then we could study driver mutations and the mutations that are related to the tumorigenesis. And we could study always all the mutations that could be related to cancer. So what challenges we have in treating cancer? Every tumor is different, and every cancer patient is different. So we have a lot of heterogeneity. For example, we could have inter-tumor heterogeneity. So in the same person or different person, depending on where is located the tumor, we could find different kind of mutations. And then we could have intra-tumor heterogeneity. This is more a more difficult issue, because we could find in the same tumor in the patient different mutation, because it could be the different cell clones in the tumor. So for example, if we take a tissue biopsy in a patient, this tissue biopsy could not represent all the landscape of mutation that that tumor has. And then we could find intercellular heterogeneity. So we could find many cells with different kind of mutation in the same tumor. For example, in this graphic, you see how we have here a normal liver, and they go to a carcinoma. So when we pass in these stages, the quantity of mutation that we found are more and more and more. So with the recent innovative sequencing techniques that we have, we could sequence genomes in a short time. For example, with the Illumina HiSeq 2000, we can read equivalent to 6 human genomes within 2 weeks. Okay, it's very, very rapidly. But the speed is expected to increase with the future techniques. So now I'm going to talk of variant classification. We have the difference with the driver mutation and passenger mutation. The driver mutation have a functional role in malignant transformation, so they play a key role in the tumorigenesis. And the passenger mutations are neutral. We don't find a functional role in tumorigenesis, but they are present in some patients. Then we could talk about 2 kinds of variants: germline variants and somatic variants. Germline variants could cause hereditary cancer because the mutation in this occurs in the germ cells of the patient. So this can be transmitted to the next generation. Germline mutation play a key role in inherited genetic syndromes. And somatic variants, the alteration occurs after conception, occurs in any cells except germ cells. Because it's not in germ cells, this can be transmitted to next generation, and this causes the majority of cancer. Then we could talk about the classification related with the effect that the variants have. And for example, we could have pathogenic variants, and like pathogenic variants, these variants are related to the tumor we are studying. And we could have variant of uncertain significance. As you see, this is the majority of variants because we still don't know the function of many variants or many genes that we found. And we don't know if that variants are pathogenic or are benign, so we classify in this big group. And then we have likely benign variants and benign variants. So you can see in this figure, for example, you can see the mutations that the gene that we could have altered in solid tumor. For example, BRAF. BRAF is mutated in melanoma, in colorectal cancer, in ovarian cancer. So there are many genes that are common in different kinds of cancer. Now I'm going to focus in breast cancer. Breast and ovarian cancer have been recognized as heterogeneous diseases comprising several molecular subtypes, each one with an associated risk profile and a specific training recommendation. The majority of cases are sporadic, but we have 5 to 10% that have germline variants, so they are hereditary. Patient with this kind of tumor is defined as hereditary breast and ovarian cancer syndrome, HBOC. Latin America is a region encompassing 33 countries with diverse culture, we know, geography and ethnicity. The resources, healthcare systems, and socioeconomic status are highly diverse among countries. And genetic studies have shown a history of migration to this region, combined with a high diversity of native population per country and impoverished population bottlenecks. So we could find in our countries founder mutations in different kinds of disease because we have high diversity, as I thought. LA is considered the most genetically mixed population in the world, and it's a specific combination of genetic ancestry may impact on the genomic determinants of the disease. So I have conducted many diseases, different than cancer. For example, I work in a study of deafness and genetic deafness, and we found a founder mutation, a Mayan founder mutation in Guatemala, because we have a lot of indigenous people and Mayan ancestry. So this group we find in different diseases, some in cancer too, and as I told you, in deafness and diabetes and other kind of diseases. In Latin America, breast cancer is the most common malignancy and the leading cause of cancer-related mortality among women. By country, the highest incidence rates of breast cancer by age are present in Argentina, Brazil, Uruguay, and the lowest in Bolivia and Salvador. So now we're going to talk about germline variants in breast cancer. So I will talk about 2 regional studies that I have participated. Okay, I will start with some genes that are related to HBOC syndrome. So germline variation in BRCA1 and BRCA2 genes is responsible for up to half of hereditary pathogenic variants in SBOC. However, germline variants in other recognized SBOC susceptibility genes have been identified, including ATM, CDH1, CHEK2, PAL2, PTEN, STK11, and TP53. Recently, germline pathogenic variants in other genes present in the same DNA repair pathways as BRCA1 or BRCA2 have been identified. For example, RAD51C, BRIP1, BART1, MRE11A. And also we can find mutation in some other DNA repair mechanisms such as non-homologous and joining. Other genes that are implicated in this syndrome is the Fanconi genes that are implicated in Fanconi anemia pathways. And this is all the genes that we can find mutated in this syndrome. And then we could have a mutation in the mismatch repair system. These are the genes that are related with this system, and the genes could cause a syndrome that is named Lynch Syndrome. Clinical, well, this is a study that we perform in Latin America and worldwide to know the status of the BRCA1 and BRCA2 sequencing in worldwide and in Latin America. So clinical testing of BRCA1 and BRCA2 began in the mid-90s, and this was only limited to 1 laboratory in the United States and a small number of laboratories in Australia and Europe. 25 years later, the number of and types of patients being offered BRCA1 and BRCA2 testing has changed dramatically due to in part the change in patent laws and increased recognition of potential benefit of testing. So high throughput sequencing technology have enabled laboratories to offer tests that cover more genes. This change in access and technology have led to a similarly intense increase over the last 10 years in the number of laboratories offering clinical testing of BRCA1 or BRCA2. So the goal of this study was to obtain a snapshot of how was the genetic testing in BRCA1 and BRCA2 worldwide. So this was the first international survey that we performed. In this first survey, they didn't include many Latin American countries. As you can see, we could see only two countries, two laboratories, one in Brazil and one in Argentina. The other was located in Europe and North America and others. Here in Europe, you can see a lot of laboratories that perform BRCA1 or BRCA2. This is in North America. So, first, in non-use labs, the equipment that is more used is Illumina MiSeq, and in US labs there is HiSeq. HiSeq is always from Illumina. The detection of deletion and duplication is performed by MLPA in the majority of the laboratories, non-use laboratories. And in US laboratories is MPS. This is massive parallel sequencing. Okay, of the 78 non-use laboratories responding to this survey, 6 report Sanger sequencing as their only method of sequencing, and 27 additional laboratories use Sanger as at least one modality. Mostly use Sanger sequencing to confirm the variants that they found. 79% of non-use laboratories reporting using both technology, using gene panels. They use gene panels to detect BRCA1 and BRCA2. It detects more than 10 genes, but 7 laboratories only study BRCA1 and BRCA2. All use laboratory reporting offer gene panels and have panels available that include more than 10 genes. Every laboratory, all the laboratories include all coding regions in BRCA1 and BRCA2. Only 6 laboratories, non-US laboratories, reported doing full intronix sequencing. And as you can see in this graphic, for example, the majority of laboratories sequence from 11 to 20 base pair of intronic regions, is a little bit of the intronic regions, because this is the mutation in this region could be related with changing the alternative splicing, and this could change the expression of the genes and the protein that we have. Then the 30% of the laboratories also sequenced the UTR's regions, 5 and 3 UTR's regions, and that was the non-coded region that the laboratory sequenced. So sequence variant was identified over half of non-US laboratories, and 75% of US laboratories confirmed variants of uncertain significance, pathogenics, and likely pathogenic by the method. The method that always the people use is standard sequencing. 27% of non-US laboratories and 29% of US laboratories confirm only pathogenic and likely pathogenic variant. They don't confirm the variant of uncertain significance. As I told you, the method that is most common use is standard sequencing. And MLPA used to confirm duplication and deletion variants. For example, in this table, you can see these are the average read depth and median read depth in non-use laboratories and use laboratories. Here is specifically for BRCA1 and BRCA2, and here is all the genes of the panel. For example, here in non-US laboratories, we have an average of 476, and a median of 758. And in US laboratories, but they have a little change, but not too much. Almost half of non-US laboratories and 30% of US laboratories report using American College of Medical Genetics and Genomics guidelines, the ACMG, for interpretation of the variants. Interestingly, more than half of the responding non-US laboratories had not specifically calculated the VUS rates for BRCA1 and BRCA2, and the ones who determined the rates reported from 3 to 50% of the presence of variant of uncertain significance. Then when we look this paper, we say, okay, Latin America, only 2 laboratories, so it's not a real survey. So we participate in this study to survey laboratories in Latin America and to complete the previous paper that was published. So we reached out 14 laboratories in 11 countries. These laboratories were choosing of those we were aware that perform complete in-house clinical grade sequencing BRCA1 and BRCA2. That means complete testing, including that the laboratory perform that DNA isolation, sequencing, data analysis, and sending out clinical reports. So this is some of the laboratories that are located in LA and LA and perform BRCA1, BRCA2 test. So A laboratories use one or more next-generation sequencing platforms to identify BRCA1 and BRCA2 variants, and only one use sandwich sequencing. All laboratories sequence all coding regions, but the intermediate regions integrate vary from 1 to 50 base pair, and 6 of 9 laboratories sequence the UTR regions. The laboratory is using NGS, all conducted panel testing, of which both BRCA1 and BRCA2 were evaluated. One laboratory includes only BRCA1 and BRCA2 in its panel, and the majority of labs perform a panel test. Here you can see in this table the countries that participated in this study: Argentina, 2 laboratories in Brazil, Chile, Colombia, Costa Rica, Guatemala, Mexico, and Uruguay. And the majority use Illumina, and only one in Brazil use Scientory. Per quality control metrics, all laboratories confirm any pathogenic and likely pathogenic variants, primarily by sandwich sequencing. 5 of 9 laboratories also confirm variants of uncertain significance, and the average read in BRCA1 and BRCA2 by NGS was 1,716x. All 9 laboratories perform variant classification in-house. 80 of 9 laboratories use American College of Medical Genetic Variant Classification guidance. Most laboratories have a board-certified molecular geneticists responsible for variant classification, whereas 3 use individual with clinical genetics expertise, and 1 use both genetic consultants and clinical genetics experts. And 6 of 9 laboratories share the variant information with public database as ClinVar. One limitation of this study is that there was no representation for many Latin American countries. We now know that we have a 64% of response rate and had a representation for 40% of Latin American countries. And now I'm going to talk about the consortium we have in Latin America. The name of the consortium is LACAM. It's a network of collaborators and cancer specialists in LA join common efforts to create the Latin America Consortium for SBOC LACAM. The LACAM study aims to recruit a large series of 3,000 cases from multiple centers in 4 years, which will make it the largest studies of SBOC in this population. So we use this panel, gene-read cancer predisposition by QIAGEN, that targets 142 genes associated with inherited cancer. The laboratories were sequencing MiSeq instrument to reach an average theoretical mean coverage of 80%. Variant calling was done with haplotype color from Broad Institute, annotation with an over and interbar. Pathogenic variant description was done following Human Genome Variation Society nomenclature, and variant classification follow the American College of Medical Genetics Genomics guidance. In this table, you can see that 47% of our patients was from 18 to 45 years, so relative young, because we know that the hereditary cancer, breast cancer, for example, is the most common in people in 40s or 30 years, or people are young. And we could see that the majority don't have a smoking history and alcohol history, and 57% say that they have family history of cancer. So 20% of the total number of pathogenic variants were found in BRCA1 and 29% in BRCA2 in the 4 countries. Patients from Guatemala and Argentina had the highest country proportion, BRCA1 40%, BRCA2 71%. Pathogenic variants in non-BRCA genes were found in 12 genes, including those in other high and moderate risk, such as MSH2 and 6, and PAL2 and MTYH. We detected a total number of 132 VUs, representing a prevalence of 41%. These variants were distributed in 76 genes, including high-risk HBOC genes like as BRCA1, BRCA2, APC, CDKN2A, CDH1, MSH2 and 6, and moderate risk, ATN, CHEK2, RAD51C, and PAL2. So this is the graphic that summarizes this study. These are the genes that were found mutated in this study. And here are the countries by color, pink and blue, Guatemala. And we have here the type of alteration that we find, if it was a stop gain, deletion, SBE, insertion, splicing. And the deletion is common in Mexico. Here we have the percentage of mutation that we have found in every genes. The majority of mutations are located in BRCA1 and BRCA2.
Claudia, sorry to intervene. We have like 1 to 2 minutes to finish.
Okay, I will finish it now. Okay, and finally, this is a study I published in the American Society of Human Genetics. Here, this was the first study, and we showed the most common mutation in Guatemala population. Here, for example, this is a splice site mutation. It was common in Guatemala, in BRCA1-2, and the deletion TT in 5 patients, and in BRCA2, a deletion of 1 in. It was the most common. So this was the first report in Guatemala population from variants, BRCA1 and BRCA2 variants. And this was another abstract that we published in American Association for Cancer Research. This we report the most common syndromes in Guatemala. For example, we found a Lynch syndrome in breast cancer with MSH6 mutation, a Lynch syndrome found related with TP53 mutation, and this syndrome Peutz-Jeghers in a young woman with pancreatic cancer, and this familial adenomatosis polyposis in colorectal cancer. This boy is 13 years old. This is rare because colorectal cancer is most common in adult people. So, but he has a colorectal cancer and mutated APC. So I think I'm gonna leave this talk here. Thank you, and I will go to take some questions.
Perfect. Thank you very much, Claudia. We've got a few questions here. I'll get one of them. Hold on. There. Victor from Yunnan asks, how does intra-tumor heterogeneity affect the manner in which cancer is treated?
Okay. It's very difficult to treat a cancer with intra-tumor heterogeneity because you could have in the same tumor, for example, in this part a kind of mutation, and this part other kind of mutation. And then, for example, if you take a tissue biopsy and you take it here, you only have the mutation that are located here, but here you don't have it. And maybe you could, the physician give a treatment to responding to one clone of one cell clone, and so the tumor is not responding well. So it's difficult because some scientists prefer, but we have to standardize more. For example, using a liquid biopsy, because liquid biopsy, we think that if we take the sample from blood, we could have all the landscape of the tumor. But this is still many physicians don't like to use liquid biopsy because we have to standardize more the liquid biopsy before we can use it. For monitoring mutation, we could use liquid biopsy, but for detect early variants, it's maybe we couldn't get it with a liquid biopsy. Okay, I think
it looks like
he
Pendleton
lost
is run into some connection issues. Yeah. So I'm going to step in. Second question we have is what kind of method did you use to calculate the risk associated with genes, and how was this risk later used to stratify genes in high and low?
Yes, this is we didn't perform this risk stage, so it is reported yet in other studies of HBOC syndromes what genes are classified as high risk and medium risk and moderate risk. It is based in the mortality of the breast cancer could cause to the people. So if they have a high mortality or a high penetrance, they are classified as high risk, and if they not too much lethality, moderate risk or low risk. So it's a classification already done